Lippincott's Illustrated Reviews, Biochemistry, Series Edition Richard A. Harvey. Contents Unit 1 Protein Structure and Function. Chapter 1 Amino Acid. Chapter 2 Structure of Proteins. Chapter 3 Globular Proteins. Chapter 4 Fibrous Proteins. Chapter 5 Enzymes. Intermediary Metabolism, Biogeneric and Oxidative Phosphorylation. Introduction to Carbohydrates, Glycolysis, tri- Tricarboxylic acid cycle, Gluconeogenesis, Glucogen metabolism, Metabolism of monosaccharides and disaccharides, pentose phosphate pathway and NADPH, glycosaminoglycans, proteoglycans, and glycoproteins. Unit 3 Lipid metabolism. The previous one was intermediary metabolism, and the previous one was protein structure and function. Unit 3 Lipid metabolism. Metabolism of dietary lipids, fatty acid, and tricyclic metabolism, complex lipid metabolism, cholesterol and steroid metabolism. Unit 4 Nitrogen metabolism, amino acids, disposal of nitrogen, amino acid degradation and synthesis, conversion of amino acids to specialized products, nucleotide metabolism. Unit 5 Integration of metabolism, metabolic effects of insulin and glucagon, the feed fast cycle, diabetes mellitus, obesity, nutrition, vitamins. Unit 6 Strong and storage and expression of genetic information, DNA structure, replication and repair, RNA structure, synthesis and processing, protein synthesis, regulation of gene expression, biotechnology and human disease. Unit 1 Protein Structure and Functions, Amino Acids. Overview. Proteins are the most abundant and functionally diverse molecules in living systems. Virtually every life process depends on this class of molecules. For example, enzymes and polypeptide hormones direct and regulate metabolism in the body, whereas contractile proteins in muscles direct contraction proteins in muscle permit movement in bone, and protein collagen form a framework for the deposition of circulum phosphate crystals, acting like steel cables in reinforcing constraint. In the bloodstream, proteins such as hemoglobin and plasma albumin, shuttle molecules essential to life, whereas immunoglobulins fight infections, bacteria, and viruses. In short, proteins display an incredible diversity of functions, yet all share the common structural feature of being linear polymers and amino acids. This chapter discusses the properties of amino acids. Chapter 2 explores how the simple building blocks are joined to form proteins that have unique three-dimensional structures, making them capable of performing specific biologic functions. Structure of amino acids. Although more than 300 different amino acids have been described in nature, only 20 are commonly found as const- constituents of mammalian proteins. Note, these are only amino acids that are coded by DNA and generic material in cell. Each amino acid except for proline, which has a secondary amine group, has a carboxyl group, primary amine group, and distinctive side chain bonded to the X carbon atom. An amino acid plays in a protein. It is therefore useful to classify the amino acids according to the properties of their side chains, that is, whether they are non-polar, have an even distribution of electrons, or polar, have an even distribution of electrons, such as acids and bases, figure 1.2 and 1.3. Amino acids with nonpolar side chains. Each of these amino acids has a nonpolar side chain that does not gain or lose protons or participate in hydrogen or ionic bonds. The side chains of these amino acids can be thought of as oli or lipid-like, a property that promotes hydrophobic interactions. Location of nonpolar amino acids in protein. In proteins found in aqueous solutions, a polar environment, the side chains of the nonpolar amino acids tends to cluster together in the interior of the protein. This phenomenon known as hydrophobic. Figure 1.2. Classification of the 20 amino acids commonly found on proteins according to the charge and polarity of their side chains at acidic pH is shown here and continues in figure 1.3. Each amino acid is shown in its fully protonated group of their nonpolar amino acids are similar to those shown to glycerin. Figure 1.3. Classification of 20 amino acids commonly found in proteins according to the charge and the polarity of their side chains in acidic pH. Side effect is the result of hydrophobicity of nonpolar R groups, which act much like droplets of oil that cluster in an aqueous environment. The nonpolar R groups thus fill up the interior of the folded protein and help give its three-dimensional shape. However, for proteins that are located in a hydrophobic environment such as a membrane, the nonpolar R groups are found on the outside of the protein, interacting with the lipid environment. The importance of these hydrophobic interactions in stabilizing protein structure is discussed upon page 19. Sickle cell anemia, a sickling disease of red blood cells, results from the substitution of polar glutamate by nonpolar valine at the 6th position of the beta subunit of hemoglobin. 2. Proline. Proline differs from other amino acids in that proline side chain and alpha amino and from a grid 5-membered ring structure. Proline then has a secondary rather than a primary amine group. It is frequently referred as the amino acid. The unique geometry of proline contributes to the formation of the fibrous structure of collagen and often interrupts the alpha helicins found in the globular proteins. B. Amino acids with unchanged polar side chains. These amino acids have zero net charge in natural pH. Amino acids with uncharged polar side chains. These amino acids have zero net charge at neutral pH, although the side chains of cysteine and triosine can lose a protein at an alkyne pH. Serine, theanine, and tyrosine each contain a polar hydroxyl group that can participate in hydrogen bond formation. The side chain of asparagine and glutamine each contain a carbonyl group and an amide group, both of which can also participate in hydrogen bonds. Diffused bond. The side chain of cysteine contains a sulfohydryl group, which is an important component of the active site of many enzymes. In proteins, the SH group of two cysteines can become oxidized to form a dimer cysteine, which contain an covalent cross-link called a disulfide bond. A further discussion of disulfide bond information. Many extracellular proteins are stabilized by diffuse bonds. Albumin, a blood protein that functions as a transporter for variety of molecules, is an example.